Welcome to the ILF 2023 and our first Industry Live of the day. Thank you so much for joining us from all around the world. We're well aware how busy you guys are, so your time is massively appreciated on behalf of me, Professor Dhar, and the team here. So today's event is called New Trends in Decongestive Therapy for Patients with Breast Cancer-Related Lymphedema, and our fantastic speaker, who's here live from India, is Professor Anita Dhar. Hello, Professor. Can you hear me okay?
Hello, Rasnik. How are you?
I'm very good.
From New Delhi, India.
Oh, fantastic. You know, very good. It's quite hot here in Nottingham, but as I speak to you and underneath me, we have a fantastic event with hundreds of delegates from all around the world. So it's very exciting times, and thank you so much for joining us today. So ladies and gentlemen, my pleasure, my absolute pleasure. Ladies and gentlemen, as you can see, we are live. So if we have any technology problems, please bear with us and we'll sort them out as soon as possible. And please engage with the event. Let us know where you're watching from, make comments, but also please ask any questions, and we'll endeavour to answer them towards the end of tonight's event. Before I start, just a massive thank you to our partners for this event, who are THUASNE. THUASNE, without your passion and commitment to independent education, these events simply could not and would not happen. So a huge thank you from me and my team. So without further ado, it gives me great pleasure to introduce you to today's speaker. Over to you, Professor Dhar. Thank you so much.
Hello, friends. Greetings from All India Institute of Medical Sciences, Department of Surgical Disciplines, New Delhi, India. We know the breast cancer has many morbidities along with it. Before surgery, after surgery, and the well-known fact is that breast cancer-related lymphedema is actually there, and it happens in 20 to 40% of the ladies who undergo surgery. And whether it's a full axillary lymph node dissection or if it's a sentinel lymph node biopsy, still there are certain percentage of ladies who will have this morbidity. Radiation therapy again plays a very important insult to the lymphatics and leads to further deterioration of the lymphedema. There are certain chemotherapy drugs that plays a role in establishing lymphedema and worsening as compared to other selective chemotherapy drugs. When we come to the general factors, it's the body mass index of more than 30 that is known as a factor related to lymphedema gain. And there are various trials where people have seen that infection, the SSI, or the cellulitis that follows after the surgery may be a reason for development or worsening of the lymphedema. So we have conducted certain trials where we found ladies who had cellulitis in their follow-up, they had worsening or onset of lymphedema. So we have conducted certain trials, we have introduced certain factors where we could prevent it. I'll share it with you later on. So we know that the medical therapy and the surgical therapy goes and is well established for the treatment of breast cancer-related lymphedema. The intervention or the non-surgical part is complete decongestive therapy, which is a well-known modality for years and years. And it is more true for intensive phase where it gives significant relief to the patient. Whereas the surgical intervention is equally important, lymphaticovenous anastomosis, nodal transfers, silicone tube placements for drainage of lymph. We have some trial done for it, and focused treatment that will take care of the cellulitis or SSI that will be useful for such preventions have been used. And we very well know various bandaging techniques and the materials that have been used in various trials and very much existing in practice these days have been established facts that take care of the lymphedema prevention as well as preventing worsening of the. So here is a trial that we introduced one of the materials and we used it against a control. Control has been the one that is being practiced in India right now. And against that, we chose Mobiderm compression bandage. The title is Safety and Efficacy of Mobiderm Compression Elastic Bandage during intensive phase of decongestive therapy in patients with breast cancer-related lymphedema. It's a randomized controlled trial and is published in Lymphatic Research and Biology in 2022. The inclusion criteria were 18 to 60 years of age and all the patients who gave consent for breast cancer-related lymphedema treatment. We excluded patients who had primary lymphedema and also who had swelling related to cardiac or any arterial disease, and also patients who were under other trials in less than 3 months prior to this. And of course, people who were not ready patients, they were excluded from the study. This was a randomized controlled trial, and all the global clinical practices were brought into practice for this. And we took our Institute Ethics Committee permission to conduct this trial. And written informed consents were taken from all the patients at the time before enrollment. So complete medical history was recorded and examination was thoroughly done for all the patients. All the baseline characteristics were recorded. Lymph volume was measured. How? I'll tell you in few minutes. Relevant exercises, the way we have established our protocol here, were taught to them and our physiotherapists practiced it along with them. Skincare, hygiene, that forms a very important management part of treatment of lymphedema. We know there is stasis of lymph and it predisposes the patient for a lot of secondary infection, and that needs to be taken care before we start bandaging them or we go for an intensive phase therapy. We have started regularly giving these patients injection benzathine penicillin via deep intramuscular route of 1.2 megaunits every 3 weeks. It's with the concept that either it's during the surgery, certain people have cultured the seroma and they found they have some element of bacteria in that. Patient may not have SSI, patient may not have fever or raised counts, but there is some element that goes with the dissection part. And second, to prevent the cellulitis and the lymphangitis that comes along with the lymphedema later on. We give it for 2 years on all the patients. The definition involved in this have used that more than 200 ml cc difference between control, that's the normal limb, and the opposite limb where the breast surgery has been done. More than 200 cc's difference was selected as definition, and for the circumferential measurement difference, more than 2 centimeters. We used water displacement method, and this was a water-filled chamber that we used. The lady was asked to immerse the hand inside a still water column with an outlet that will be collected in a measure that is a linear measurement scale in the measuring cylinder that is kept at the outlet to control or capture the water that is displaced by the hand and measure it. That will be equivalent to the volume that the hand and the arm of this lady had. That's the water displacement method. That's a gold standard, and we use this method for this. And the 2 readings were at the baseline before the surgery and 15 days after the compression bandaging was done for comparison. This was a randomized computer-generated code, 1:1 ratio. So there were 2 groups. One was the Mobiderm group and another was the control group. Multi-component compression bandage regimens were used. The first, that is the inner and the outer layer, were the same in both the groups. That is, the inner layer was cotton bandage, band, and the outer was the elastic short stretch bandage. And the middle part was the only difference between the Mobiderm and the control. The middle part or the intermediate layer was the Mobiderm band by the THUASNE company, and that was used for the as the intervention group. We have a specialized certified lymphedema therapist in AIMS who was involved in the treatment and introducing the bandaging for these patients, how to do it, what's the technique, which was introduced to the patient so that that can be reinforced 2 to 3 times, 3 times in the first week, and then 1 time in the second week to check whether the patients are doing it properly at home or not. The outcomes were, primary outcome was the reduction in the volume measured with the water displacement method. And secondary outcomes were these patients have pain, and the worst thing is morbidity because of the heaviness that the stasis of the lymph brings along with it. The limb becomes so much heavy that lifting and using it for day-to-day activities, a simple activity like combing the hair, becomes a big issue. That was recorded as a secondary outcome. There was no prior study that could tell us any difference available with the use of Mobiderm band. So we used an arbitrary number that was 50 for against the sample size calculation. 25 in each group, Mobiderm and control group. One patient assigned to the control group had to withdraw due to some personal reasons after the first visit. So we had total of 49 patients as the results. So this is the baseline characteristics of the patient, where we have selected age, BMI, any comorbidities that can bring again trouble for this patient, what kind of surgery was done, what kind of disease this patient had, whether when the lymphoscintigraphy, which is a routine protocol for our workup, whether patient had no block, partial or complete block, and the surgical intervention, whether the patient has seroma, infection, and they were all recorded, and we found there was no difference in the baseline characteristics. They were matching well. This table reflects the volume changes that have happened in the 2 groups. One is the absolute limb volume measurement, and second is the excess volume that we calculated, and we found that between the 2 groups, there was significant difference. And within the groups, of course, there was. And we had to use unadjusted mean and also the adjusted mean. We realized that the difference between the Mobiderm and the control group was not showing up in an unadjusted manner. What was the difference? The difference was the control. The control in the opposite limb was different in many patients. For example, a lady who is little overweight may have a different limb. So you cannot head-on compare mean to mean. So we had to do the baseline adjustments, and this is the adjusted mean group, which showed actually a significant difference between the 2 groups. It was 0.003, and in the adjusted limb value, absolute limb volume, and also in the excess volume group, 0.006. That was evident in our clinical practice also. And this is the figure showing such volumes and how the changes took place over 15 days. And this is with the excess limb, how the volume decreased over a period of 15 days. The intense treatment from 0 to 15 days was recorded. This is the volume reduction. Volume reduction is much more with the Mobiderm group as compared to the control group. When we look at the overall picture of the volume, the reduction, we can see that affected limb reduction, how it changes. There was overall 92.5 to 421.2 ml difference. That was quite significant. In the volume reduction, again, it was 69.3 to 371.3. That was again 0.006, favoring the Mobiderm group. When we come to record the VAST score, visual analog scale, we found the pain and heaviness is significant between the mobiderm and control group favoring mobiderm. People were happier with a much more reduction in the volume, much more reduction in the heaviness of the limb with the mobiderm group. So multi-component compression bandaging system is key component in lymphedema treatment in both phases, and in particular during the intensive phase therapy. Although different modalities exist, a combination of layers of inelastic or low stretch compressive bandages and short stretch elastic bandages applied to the whole limb are commonly used. Long stretch also have been used in combination with multi-layer bandage. What about the mobiderm mobilizing pad or the bandage? It's designed to apply different areas of pressure on the skin surface, which is in combination with muscle movement, promotes edema, mobilization, and therefore evacuation. Mobiderm use has been tested in various clinical studies for maintenance phase of lymphedema treatment. Data from clinical studies indicate that the use of mobiderm garment also during the night time has been helpful in such patients for benefit in BRCL. How does it work? It has foam blocks of different sizes, 5 to 15 millimeter on each side, inserted between two non-woven layers of material. The mechanism of action of the mobiderm combined with elastic compression or non-elastic support exerts differential pressure on the supporting zone and its surrounding environment or the area. This combined with variation in muscle volume creates a shear effect on the thick, solid, indurated subcutaneous tissue. That is what is the definition of lymphedema. So it exerts direct impact on such and gives its benefits. These are examples of some of our patients where we see how it has been beautifully applied on one of the limbs. She was a breast cancer patient, operated, and axillary lymph node dissection was done. This is how they recover. And this is one more patient, the lymphedema, how the bandage has been applied, and the good results and the happier results this patient had after that. This is one more example. You can see the difference. The volume reduction happens, the heaviness decreases, gives much more better quality of life to the patient. So CDT, I conclude my talk by saying the CDT forms an important part of lymphedema treatment. Combining mobiderm with other multi-layer bandages is associated with a shorter period of decongestive lymphedema therapy as compared with other bandages, suggesting a rapid effect on reducing edema and improvement in pain, heaviness, as proved by our improvement in the VAS scores, have been really beneficial to the patients over a conventional or a control group. Therefore, this bandage combination would help us set a new trend in improving the quality of life of such patients, which means a lot when we see these distressful patients in our follow-up clinics in breast cancer. Thank you so much for your attention. I may be pleased to answer any questions from the floor. Since it was a short duration treatment, they welcomed it and happily got themselves bandaged. And see, the problem is that they are really distressed with the lymphedema, and directly there is hardly any treatment. We can't. No one in the globe can actually say that promise, I'll just cure your lymphedema. There's nothing happening as of today, and whatever best treatment they receive and it works for them, they are there. They do come and they follow up. And yes, after they are treated and they are happy with the reduction, they may have less visits. But in this, at least in this trial, so for one patient, they did come and meticulously bandage themselves, and they try for the learning process also, because we call them for different visits, and it was a good experience. Yes.
Fantastic. That compliance and engagement is so important. So question 2.
What happens, the limb
Sorry.
becomes very heavy. It depends on the duration, and the volume is the measurement part, the outer size. But due to the water content or the lymph content of the inside the limb itself, it's very difficult for them to lift it and use it for combing. And a housemaker who is cooking and doing a lot of things, activity at home, it means a lot. And when this water content lymph, it comes down, the lymph it becomes lighter. Even if the girth is a little bigger, it doesn't matter too much. So they are looking for a more manageable limb than the size. Size of course comes down once the fluid comes out, but they are looking for a lighter limb.
Fantastic, thank you. So we're going to move on to question 2. Were other bandages allowed in the study beside the ones that you've mentioned?
No, we just had 2 groups, and as we mentioned in this trial, 2 arms, 3 layers in each, and the only difference was the middle layer, which was replaced by the Indian-made layer in the control group. No other bandages were allowed.
Fantastic, thank you. So the next question is, Professor Dhar, you've spoken about the water displacement method. Is this a method you use regularly?
We use this especially for, it's supposed to be a gold standard method for measurement of the volume, but there are other methods available. Indirect methods like pyrometry, they measure through infrared rays the girth, and then they calculate with the formula and take out the volume. And then there is bioimpedance spectrometry. They use a spectrometer and quantify the fluid. It sends in a very low current across, and the more the fluid inside, the more the lymphedema, the resistance is lower, and that is calculated then as the fluid content. And there are latest techniques that are now used for a long time. I should not say latest, but indocyanine green, for example, is injected in the web, and we see the flow, and this helps us in comparison. In fact, we have conducted one trial where we picked up all the patients immediately before and after surgery, wherever we have done an axillary dissection, and we found that this was a very good modality to identify a delay in passage of this indocyanine green. It used various patterns, the stardust and the splash and all.
Yep.
Linear is when it flows very nicely, the lymphatics are patent. So you can indirectly judge whether there's lymphedema and what degree it is.
Fantastic. So we have 3 more questions, and then we'll leave you to your busy day ahead. Do you use other methods to quantify lymphedema reduction in your daily practice?
We use the water displacement method actually. This is for trial and some select cases, but we use the girth measurement, that is with the select special tape that at various distances from fixed bony points. As per the guidelines, we use the girth measurement. That's quickly done in the clinical setup, and we measure the diameter actually.
Fantastic, thank you. So question 5, and thank you for all the questions that keep coming in. Were there any severe adverse events, and were they different from what you expected?
Not in this trial. So we take precautions. We have a protocol-driven practice, and before we start compression in upper limb or in lower limb in lymphedema cases, we have a protocol of getting our Dopplers done, both for venous and as well as the arterial Doppler. See, the complications come up if we have not evaluated the limbs properly. For example, if this patient has arterial disease also, and we're giving a compression on it, definitely patient will have compromised circulation and we're worsening it. So we do take care. It's a protocol that we do Doppler, arterial and venous compulsorily for these patients. And second thing is, very strict instructions are given that in case there is pain or discomfort, they should open the bandage immediately. And one family member other than patient is also a witness to the bandaging technique, and that's the precautions we take. And we had no adverse event or SAE in this trial.
Fantastic, thank you. So over to our last question. What do you consider to be the major strength of your study?
We very meticulously followed our randomized controlled trial and inclusion exclusion criteria. We followed our volume measurements very meticulously, and there was no compromise on that. And head-on compared, we did see, as we have put in the table, adjusted and unadjusted, both because of our, we have a very reputed, highly reputed biostatistics person. He is an experienced person, and he suggested that we should adjust it patient to patient. So it's not a head-on comparison of the mean to mean, that will not help. So every patient, the volumes were individually compared from baseline to the opposite side. Like it was 1:1 comparison, not mean to mean. So there are strong points from the biostatistical point of view, there are strong points from the clinician's point of view. Both have been taken care in this study. No compromise.
Fantastic. Listen, Professor Dhar, thank you so much for your time today. And that concludes today's event. A massive thank you to everyone out there watching. A massive thank you to you, Professor Dhar, not just for the last half an hour, but also the preparation you put into this presentation in the weeks and months prior to this. A big thank you to...
So much happy. It was a pleasure. I wish I could have been there, physically present and enjoying the conference and as well as this presentation.
Listen, thank you so much. A massive thank you also to THUASNE. As I said before, that commitment to independent education and product innovation is key to this specialism moving forward. A thank you to the digital team.
And a special thanks to THUASNE for helping us in this trial, and it was a great pleasure working with the team. Thank you.
No, thank you. As I said, you know, the commitment from, oh no, absolutely, thank you. The commitment from industry, from THUASNE, you know, drives innovation in this space, and it's fundamental, I think, moving forward. A big thank you to the team here. Thank you so much for your time. Please stay safe, stay strong, and we look forward to seeing you at future events. Thank you so much.
Thank you.
Thank you very much, Professor.